Hello guys, my name is Dr. Varun and in today's video we're going to study about another very important concept from the chapter number 2, that is spermatogenesis. Well, the name itself is telling us what we're going to have to study about in this chapter. So spermato word is coming from sperm and genesis means formation. So the chapter is dealing with the two things, that is sperm formation. So first of all, you should know that location of the event, where is it is occurring? It is occurring in the seminiferous tubules of testes. Now the next thing we have to study is that how a sperm look like. We know that the sperm, like I'm not doing in the detail about it because that's a totally different topic. Here we are just studying about the basic of it. That is what, that your sperm will have a head-like structure where here the nucleus would be present. Now the next thing is that around it there is a presence of a cap-like structure called as acrosome. Next thing over here is what, at this area there is a presence of two centrioles, which is called as proximal and the distal centriole. Moving it in this region, there is presence of mitochondria which are present helically around a axis. So means if this is the axis, then where it is present? It is present around itself helically. Okay. Now the next thing is, till this structure they are covered by what? By a thin membrane, and this is the plasma membrane, and the area that is surrounding it will have what? Cytoplasm present. Meanwhile, moving on, this area after this is what? The tail is present. Okay, and end part have a axial filament. At the end region is what? End region is naked. It do not have any membrane covering around itself. Okay, so this whole structure we have to make. Okay, now the starting, it do not look like this. This characteristic shape is not there. So how it looks like? But we have studied this thing in the last chapter also, that how the different cells in the different stages of development would be present. Here we are not going into the deeper part of it. You can take the reference from that video, the last video. Here we are seeing that how the process will go and what will happen. Okay, so first of all, your spermatogenesis process which you are studying, in this, where it is occurring? Seminiferous tubules of the testes, and every day 100 to 200 million sperms are formed after the puberty till the old age. Of course, after certain period of time, the amount or the quality of the sperm content may alter with the age, illness, or the depending upon the person to person. Okay, next thing is that let's recall that what are the different stages of the spermatogenesis. So if you remember that the story starts from spermatogonia, it is changing into the primary spermatocyte, then forming secondary spermatocyte, then forming the spermatids, and finally the spermatozoa. So this only process we're gonna see now. So in this process, the first thing is during your what we say that embryonical development time, from your yolk sac. Okay, what is yolk sac? Yolk sac is a membranous covering or a structure, sac-like structures present around the fetus, which contains lot of stem cells, germ cells, and even initial days in the fetus, it helps in the formation of blood cell also. So one such germ cells are present over there. We call it primordial germ cells. Okay, they migrates to the testes. That's it. So the yolk sac has a primordial germ cells which migrates from this region and goes into the pelvic region where the testes will be present. Okay, now the next thing is that over there, these primordial germ cells are called as spermatogonia. So the first thing we have to remember, that is spermatogonia. Next thing, these spermatogonia, since the body is what, diploid, every cell is making from the zygote is diploid. So this thing would also be what, diploid in structure, which we have to remember. Next thing, what will happen, these spermatogonia will divide into two different types of the cells. One will form the precursor of the sperm cell, whereas the other one will act like what, it again goes and form these kind of cells. So we have a type A and type B cells. Now the precursor of the germ cells, what our major concern is. So precursor of sperm cell will do what, these cells will mitotically increase its number and differentiate. So if it is differentiating, means it's changing its form. So now it will be called as what, primary spermatocyte. In this primary spermatocytes, these cells are also what, all these cells are diploid only, but from these primary spermatocytes, the first process, that is meiosis, will start, because basically we are making sperms. Sperms are what, gametes. Gametes are always haploids. So these secondary spermatocytes will undergo its first meiosis. Okay, and it will form dyad of cells. Dyad of cells means two cells will be formed, and these two cells will be called as secondary spermatocyte. Secondary spermatocyte. Now, by the time these secondary spermatocytes have formed, the ploidy is also reduced because the homologous chromosomes has been separated. So these are haploid structure. Okay. Now, in the next process, since this was what, meiosis 1, it will undergo its second meiosis. Okay, so both of these cells will undergo its meiosis 2, and we call it now what, the tetrads of cells are formed. So each primary spermatocyte will make spermatid. So total there will be formation of 4 spermatid cells. All these spermatids are haploid in structure. Now, when the whole process is occurring, these cells are joined together, means they have a cytoplasmic bridges. Means if this is the primary spermatocyte, this is the primary, it is dividing into 2 secondary spermatocytes, but they are not completely divisible. There is a connection present. This connection is called as cytoplasmic bridges. Same goes with when these two will divide further. So the whole structure will appear somewhat like what? It will appear, which I am making over here, 1, 2, 3, 4. So these are joined by cytoplasmic bridges. Okay. So now what will happen? So first, after this, what will happen? These cytoplasmic bridges are broken down. If someone will ask like, why cytoplasmic bridges are present between that? What is the role of this cytoplasmic bridges? Then these cytoplasmic bridges help in the equal or the same growth among the 4. There is no unequal growth is there. So that to ensure that this thing has been maintained over there. So equal has been divided by the bridges. Okay. Now these spermatids, they are present into the area, but now they will further develop or matures into what? They all will develop into the structure called as spermatozoa. So all will make spermatozoa. Spermatozoa is also what? Haploid structure only. Now here you have to remember that the change or the development or maturation of spermatid into spermatozoa, this is called as the spermiogenesis. Spermiogenesis. This is not spermatogenesis. Spermatogenesis is this whole process. Spermiogenesis is one single step, that is from spermatid to spermatozoa. Moving on, these spermatozoa are embedded into the Sertoli cells, tight junction Sertoli cells, which we might have studied in the last lecture. So that spermatozoas which are embedded into the Sertoli cell will be released by pressure into outside from the testes. Okay, this process of release of spermatozoa from testes to the outside, okay, or from the seminiferous tubules to the outside, is known as spermiation. Okay, so the last thing that we will study, that is spermiation. So what is spermiation? Spermiation is the process of release of spermatozoa from seminiferous tubule to outside duct. Okay, that's all for this video. I hope you like this video, and you have any doubt, just put down in the comment section, or you can directly message me on WhatsApp about it. Thank you.